that. All right, class, so welcome to Tuesday night. We're in October 5th. We're into our second module. As you can see from this most recent announcement that I posted, we have a lot of fun things going on this week in class. We're going to be starting our nervous system introduction in chapter 11. And you can see that PowerPoint shown here in this announcement. What we'll do is this PowerPoint in chapter 11 is a little lengthy, and like usual, I'll kind of pick out parts that I want you guys to focus on. If we don't finish going through chapter 11 tonight, what I'll do is I'll do another recording, probably tomorrow, and post it to our class YouTube page to finish up chapter 11, just so that Thursday we can maybe start chapter 12. You will have a lecture quiz on chapters 9 and 11. Due by this Sunday, and I think I open it up on Friday for you guys. And I took off Proctorio for that quiz, and you can use your notes for that quiz. So it's a little less stressful, hopefully, for that quiz coming up. Also, you'll have your first Physio X lab, which uses the MyLab and Mastering of the textbook. And I've created a video recording about how to use the MyLab and Mastering, how to get into the skeletal muscle physiology. Yes, I know the PowerPoints are a lot of information, yeah. So I created this recording, and I'm not going to go through any of that now because I didn't want to take up a whole lot of time of our class. So look at that recording for how to do your Physio X lab. And if you still have questions about that, just send me a message, but you do have a Physio X lab due this Sunday. So a quiz and the Physio X lab, that Physio X lab might take several hours to complete. So please get started on that. So with that, let's jump right into chapter 11, which is the introduction to the nervous system. You know, I always say our textbook is an anatomy and physiology textbook. So you'll see some things that you might have learned in anatomy, but you might not have remembered all these things. So I kind of go over them quickly. And again, I'll try to pull out and focus on things that you should focus your studying on, as well as I've created some poll questions that you might see in a future quiz or exam that we'll also go through tonight. So let's talk about the functions of your nervous system first. So your nervous system, so we talked about your skeletal, your muscular system, now we're on to the nervous system. It's the master controlling and communicating system of the body. The cells of the nervous system will communicate via electrical signals and chemical signals. So your body is like this electrified circuit, and all the cells of the nervous system are communicating with each other. It's very rapid and a specific response, and usually the cause is almost an immediate response. Letting a couple other people in. All right. So basically, the nervous system has three functions that overlap. The sensory input is all of the information gathered by sensory receptors, either external to the body or internal to the body. The integration is the processing and interpretation of that sensory input. So usually what happens is the sensory neurons take in all the sensory stimuli, send it to the brain and spinal cord where integration occurs, where the brain and spinal cord figure out and processes that interpretation of the sensory input. And then there's some sort of motor output or response. And this is an activation of any sort of effector organ like your muscles and glands to produce a response. And that's a really simple way to describe the nervous system. And here we have the nervous system functions. So for example, you see a glass of water, you're thirsty, that's sensory input. Your brain interprets that, and then it tells your biceps to pick up the glass so that you can take a drink. We divide the nervous system into your central and peripheral nervous system. Your central nervous system is your brain and spinal cord. This is your integration and control center. And the peripheral nervous system are all of the spinal and cranial nerves outside of the brain and spinal cord. And this is just a division of your nervous system, the CNS and the PNS. And just kind of solidify and cement that into your brain. The central nervous system is brain and spinal cord, and peripheral nervous system are all the cranial and spinal nerves. It also includes the ganglia, which are collections of cell bodies. So yeah, I'll kind of refer to them from here on out as the CNS or PNS, just so that you are aware of that. So here are the functions of the nervous system and how we kind of divide the functions of the peripheral nervous system into two functional divisions. And I ask you several test questions based on kind of the following pages. So understanding the functions of the nervous system, we have a sensory or an afferent division. Your somatic sensory fibers will convey impulses from skin, skeletal muscle, and joints to the brain and spinal cord. Visceral sensory fibers, the word viscera has to do with organs. These will convey impulses from visceral organs to your central nervous system. So these are all sensory or the afferent division. Anything that's conveying an impulse either externally or internally to the brain. And then the motor or the efferent division is the response of the central nervous system then out to an organ, a gland, or a muscle. And we divide your motor division into the somatic nervous system, which usually goes to skin, joints, skeletal muscles, and the autonomic nervous system. These are things that happen subconsciously that are out of your control, and we'll talk about that. So your somatic nervous system, and again, if you just joined us, there will be at least 2 or 3 questions based on understanding the differences between the sensory division and the motor division and kind of the subdivisions of each. So spend some time kind of studying these slides for the lecture exam. So your somatic nervous system includes somatic motor nerve fibers that conduct impulses from the central nervous system to your skeletal muscles. And this is voluntary nervous system. So this is your conscious control of your skeletal muscles. So you're controlling this. Then the autonomic nervous system consists of visceral motor nerve fibers. So it's still a part of that motor or efferent response, but these are going to your viscera and they're under involuntary nervous control because this is happening automatically, subconsciously, without you thinking about it. And this is where we have 2 functional subdivisions, the sympathetic and the parasympathetic autonomic nervous system. And these will work in opposition to each other. And you should know well the differences between the sympathetic and the parasympathetic division. So here we have the organization of the nervous system and kind of what I just went over, and I'll go ahead and make this a little larger for you. So here we have your CNS and your PNS, and the peripheral nervous system is kind of what I want you guys to focus on in these divisions, understanding the sensory, the afferent division, the motor, the efferent division, and we divide the motor or efferent division into a somatic and autonomic nervous system. The autonomic nervous system, then we have the sympathetic and parasympathetic division. There is a test question about what's the difference between your sympathetic and your parasympathetic division. And your sympathetic autonomic nervous system, shown here, prepares your body for emergencies or activities. So this is what we call your fight or flight response, is the sympathetic division. And the parasympathetic division is the division of your autonomic nervous system that prepares your body for rest and digest. So after a big meal, Thanksgiving, the holidays are coming up, you eat a big meal. The parasympathetic division is what prepares your body for digesting the food and resting and relaxing. So then we'll get into neuroglia, which describes nervous tissue histology. So basically, we're just going to look at the cells of the nervous system now. You should know the general cells of the nervous system. And the nice thing is it's pretty simple. There's neuroglia, which are glial cells. These are just the cells that support and surround the neurons, which are the actual nerve cells that are excitable. They transmit electrical signals and communicate those signals between each other. So here are the 4 main neuroglial cells that support your central nervous system. So all of these neuroglial cells will be found in the brain and spinal cord. We have astrocytes, microglial cells, ependymal cells, and oligodendrocytes. there might be one test question about, or a matching question that describes one of these four types of supportive cells. So astrocytes, really simply put, they're the most abundant, versatile, they cling to neurons. The big thing that they do is they support and brace neurons. They participate in information processing, they control the chemical environment. They play a role in exchanges between capillaries and neurons, and that's called the blood-brain barrier that I will show you. So these are astrocytes. So this is what an astrocyte looks like. It has what we call, you can see the nucleus there, it has what we call like little perivascular feet that will wrap around the capillaries. And what astrocytes do is they try to keep harmful things inside the blood, which is inside your capillaries, from leaking out into the surrounding brain tissue. So we call that the blood-brain barrier that the astrocytes form. And that helps to keep harmful substances from going into the brain tissue. Some things can get through the blood-brain barrier, like nicotine and alcohol. Microglial cells are the cells that help to phagocytize microorganisms. So this is kind of a defense mechanism in your brain and spinal cord. They help to pick up anything foreign, digest it, and get rid of it. So this is a look at a microglial cell, kind of your, like a kind of a defensive cell in your central nervous system. And then the ependymal cell, this might be a test question, lines the central cavities of the brain and the spinal column. So you guys have, or we all have ventricles or spaces in the brain that are filled with cerebrospinal fluid. And these ventricles are lined with ependymal cells to help form a permeable barrier between cerebrospinal fluid in the cavities and the tissue fluid bathing the central nervous cells. So these are the ependymal cells lining the fluid-filled central nervous system cavities. They usually have cilia attached to the top as they're helping to kind of wave that cerebrospinal fluid around. Oligodendrocytes, again, we're still in the central nervous system. This is the fourth kind of supporting cell. These are the cells that insulate nerve fibers with myelin sheaths. So those are oligodendrocytes, and we'll talk about what myelin is. So this is what oligodendrocytes do. The myelin kind of forms like an electrical tape around the axons of neurons to help increase conduction speed, velocity, action potentials, or signals that travel through the neurons. Then we only have two major neuroglial or support cells in your peripheral nervous system. We have satellite cells, and we also have Schwann cells. And oligodendrocytes create myelin sheaths in the central nervous system, and Schwann cells create myelin in your peripheral nervous system. So be sure to keep those two straight. So they're functionally similar to oligodendrocytes, and they form the myelin, which is shown here, which again is like electrical tape that covers the axons of neurons to help the electrical signals to travel quickly down a nerve fiber. So then the neuron itself is a nerve cell, and these are the structural units of the nervous system. They're large, highly specialized cells that conduct impulses. You should know their special characteristics. They have a high metabolic rate because they require a constant supply of oxygen and glucose. They are amitotic, and that means they do not go through mitosis. So your nerve cells are not able to regenerate or divide. When a part of your nervous system is damaged, it's just damaged forever. So this is why things like paralysis or severing of the spinal cord, there's not a whole lot that can be done because your nerve cells can't regenerate. They can't go through mitosis. So they are amitotic or not mitotic. They have an extreme longevity though. They last a person's lifetime because they don't replace themselves. So they last a long time, they're excitable, and they have a high metabolic rate. They all have a cell body and one or more processes. The cell body is the biosynthetic center. I'm going to kind of go through this. Most neuron cell bodies are located in the central nervous system. The nuclei are clusters of neuron cell bodies in your central nervous system called nuclei. And ganglia are clusters of neuron cell bodies in the peripheral nervous system. And we kind of bring up the idea of nuclei and ganglia as we talk about kind of further kind of delving into the nervous system later. So here are some of the processes that form a neuron. Arm-like processes that extend from the cell body are their processes. Tracks are what we call bundles of neuron processes. And the idea of a nerve is just bundles of neuron processes in the peripheral nervous system. So in the central nervous system, for example, in the spinal cord, we have tracks, which are bundles of neuron processes like axons that run up and down the spinal cord. And then nerves are what we call bundles of neuron processes in the peripheral nervous system. So you have radial and ulnar nerves in your forearm, and they are just bundles of axons. And these are the two types of processes that come off the cell body, dendrites and axons. Maybe this looks similar to anatomy, learning the structure of a neuron. You have the cell body, the dendrites that receive the signal, the cell body receives that signal, integrates it, and then sends along the impulse down the axon. This area right here is called the axon hillock, and this is usually where action potentials begin, and we'll talk about action potentials later. And then you can see the Schwann cells creating the myelin sheaths just to help the conduction velocity of those impulses traveling down the axons. So I'll let you read through some of this on your own. Dendrites are the input region of a neuron, and you can see dendritic spines. They will receive input and send it on to the neuron cell body, which will kind of integrate it and figure out what's happening. The axon structure, it starts kind of at the axon hillock where it connects to the cell body. Long axons are called nerve fibers. And the axon is usually where the action potential will occur, which means this is where the nerve will send on its signal down the length of the axon process. So I'll let you guys read through some of these functional characteristics of the axon, and we're going to go over a lot of these functional characteristics when we talk about how they work. Something that's interesting about movement of the electrical impulse in an axon, and the electrical impulse in an axon can go away from the cell body, and this is called anterograde. This usually occurs in mitochondria, cytoskeletal elements, membrane components, and enzymes. Or the direction of the impulse can be retrograde or toward the cell body, and this usually degrades organelles, signal molecules. This is what viruses are and bacterial toxins are retrograde because the signal is going back towards the cell body. And this is interesting because certain viruses and toxins can actually damage neural tissues by using retrograde axonal transport, meaning that bacteria and toxins will travel to the cell body and damage the cell body of that neuron instead of moving away from the cell body. And this is an example is polio, which will eventually damage the nerve that gives nerve supply to the diaphragm. So it causes people to not able to breathe, rabies, herpes, similar viruses, and tetanus. Okay, so really briefly, the myelin sheath is composed of myelin, and the function of myelin is to protect and electrically insulate the axon. And what it does is it increases the speed of the nerve impulse transmission. So myelinated fibers have myelin, and non-myelinated fibers do not contain that myelin. So non-myelinated fibers will conduct impulses much more slowly. The myelination in the peripheral nervous system is made by those Schwann cells, and it looks like kind of like a jelly roll surrounding the axons of the neuron. And as we look at how myelin sheaths form, you can see how they form kind of like jelly rolls or layers of myelin sheath around the axon, which is in yellow. So here's a look at the axon of a nerve and the myelin sheath layers surrounding it, looking at an electron micrograph of about 24,000 times magnified. In the peripheral nervous system, the myelin sheath forms gaps or spaces between sheaths, and these gaps are called the nodes of Ranvier, probably named after the man who discovered them. And non-myelinated fibers are not wrapped in myelin. So one cell may contain 15 different fibers. So non-myelinated fibers will conduct impulses much more slowly. So again, here is a look at an axon covered, surrounded by those myelin sheaths. And these are the gaps or the nodes of Ranvier, where there's just a gap between sheaths. And that is important because the gaps are kind of how the electrical signal will pass through. Myelin sheaths in the central nervous system are kind of what gives the central nervous system its gray and white matter. Myelin is white in color, so white matter will be regions of the brain and spinal cord with dense collections of myelinated fibers, which are usually fiber tracts, which need to carry the impulse much more rapidly, and the myelin will help with that. And gray matter will not have those myelinated fibers because it's more gray in color. So here's a look again at oligodendrocytes. These are what create myelin in your central nervous system, the brain and spinal cord. We have some structural classification of neurons, multipolar, bipolar, and unipolar. I'm just going to go ahead and show you the picture of this. The multipolar refers to the number. Multipolar means many processes. So we have dendrites and an axon. This is what most of the neurons are in the body. Bipolar, bi for two, has kind of two processes, one dendrite and one axon coming off of the cell body. And we'll find these in the retina, in the eye. And then unipolar or pseudo-unipolar is kind of a one axon or one process coming off of the cell body. But again, most of the neurons in our bodies are the multipolar. Here's another kind of look at where they could be found. So bipolar neurons can be found in the olfactory mucosa, the eye, the ear, and you can find here where unipolar neurons can be located. I won't ask you guys where they're located. So again, functional classification of neurons, you should understand this. You can have sensory neurons or motor neurons, and sensory neurons transmit impulses from sensory receptors to the brain and spinal cord, and motor neurons carry impulses from your brain and spinal cord to effectors. Interneurons are also called association neurons, and they are just kind of the mediators between. So they lie between motor and sensory neurons, and they usually just shuttle signals through the central nervous system pathway. And about 99% of your body's neurons are just interneurons or connections between the two. And here are looks at multipolar, bipolar, and unipolar neurons and where they can be located. Okay, so before we get into membrane potential, let's ask a couple poll questions, and maybe you'll see some of these questions again. I'm not sure. So let's launch this poll. If you hopefully can see this. You guys see the poll in front of you? So the question, good, I got one response, so I know you guys can see it. Nerve impulses are sent to slow the heart's rate of contraction. The nerve fibers sending these signals will most likely belong to which division of the nervous system? And these are completely anonymous. I have no idea who's answering, so feel free to go for it. Remember that the parasympathetic division is your rest and digest division. So the division of your body that slows down the processes, it gets your body ready for resting. It sends all the blood to your digestive system to help digest food. And the sympathetic nervous system is your body's fight or flight response that gets your body ready for emergencies. So I got a couple more questions then. A couple more. Okay. So I'm going to end the poll and I'm just going to share the results with you. So most of you got it. So this is a parasympathetic division. So the parasympathetic slows your heart rate. It slows the body down. It sends all the blood and all the resources to help digest the food. The sympathetic division would help to increase heart rate and kind of get the ready body for a fight or flight response. Good. Let's do another one. All right. Nerve impulses leading to the brain carry information about cool temperatures on the skin, and the nerve fibers sending these signals will most likely belong to which division? So which division of the nervous system will take an external or an internal stimulus from the outside of the body or the inside of the body? In this case, it's cool temperature, and the nerve fibers will send these signals to the brain. So most of you guys are getting this. So I'm gonna, I think all of you, good job. So from a sensory input to the brain is always a sensory, or an A for a part of division. A somatic motor division would always be a response going out from the brain or spinal cord, telling your skeletal muscles to do something. Okay, we're just gonna do a couple more. Let's see here. This should hopefully be an easy one too, because it kind of piggybacks off the last question. Your nerve impulses leading to skeletal muscles carry information to direct movement. So they're coming from the brain to skeletal muscles. The nerve fibers sending these signals will most likely belong to which division? And as a hint here, somatic, kind of think of the S in somatic and always kind of attach that to the S in skeletal muscles. So somatic is always attached to skeletal muscles, and your somatic is also a motor division. So this would be the somatic or motor division. Good. And I think I just have one more that I'm going to share with you. Hopefully this is a nice break. Okay, we'll ask this one, actually two more, and then I'll stop. What type of cells line the ventricles of the brain? Ependymal cells, neurons, epithelial cells, or astrocytes? And this is a question, and again, you might not get a specific question on your lecture exam, but you might get one about one of these other cells. Knowing these microglial cells, whether it's an astrocyte, ependymal cell, oligodendrocyte, Schwann cell, microglial cell, you should know just the basic functions and what they each do, just in a simple question like this. So the ones that line the ventricles of the brain are actually the ependymal cells. So if we talk about, supporting cells, we're talking about ependymal, astrocytes, microglial cells. Neurons are just the nerve cells that conduct impulses, and they're the kind of the electrical signaling of your nervous system. But this is ependymal cells. And then last one, and then I'll move on. Which of the following is not a characteristic of neurons? They have an exceptionally high metabolic rate. They have extreme longevity. They conduct impulses, or they are mitotic, meaning they can divide and regenerate. And your nervous system, if it gets damaged, it can just grow a new nerve. Does that happen? And a lot of you are getting this one right too, so that's good. Okay. Looks good, and I'll just share those results. So they are mitotic is false. So your nervous system is not able, think of mitotic mitosis, kind of dividing, creating more cells. They are actually amitotic, the slide said. Okay, so let's skip the polls. Hopefully you're not seeing anything. Okay, so a membrane potential, and we'll get through a couple more slides, and then what we don't get through, I might finish in another recording. And again, please watch the majority of these lecture videos, because these PowerPoints in the nervous system are long, and I skip over some of the information. So membrane potentials, we talked about the neuromuscular junction and membrane potentials and depolarization, repolarization in muscle cells, to kind of send along an action potential in a muscle cell. The same kind of thing happens in a nervous cell. And one of your workshops is on action potential. And again, this is kind of a shout out to make sure you're going to your workshops. The action potential workshop will be very helpful in this module and for this exam. So like all your cells, neurons have a resting membrane potential. And unlike most other cells, neurons can rapidly change their resting membrane potential, which makes them highly excitable or able to respond to a stimulus. So what do I mean by that? Let's talk about some basic principles of electricity. First of all, opposite charges are attracted to each other. Energy is always required to keep opposite charges separate across the membrane of the axon of a neuron. Energy will be liberated or freed when the charges move toward one another. And when the opposite charges are separated, the system has potential energy. So we use this idea of voltage, which is a measure of potential energy generated by a separate charge. And in neurons, we measure the voltage between 2 points in millivolts. And this is called the potential difference or the potential. And this is just the charge difference across the plasma membrane. And the greater the charge difference between the points means there's a higher voltage. Current is the flow of electrical charge, and in terms of the nervous system, ions will flow across the membrane. It can be used to do work, and it's dependent on voltage and resistance. And I won't get too much into Ohm's law or defining kind of the relationship between current, voltage, and resistance, because you can still understand the concept without us going that detailed. So the role of our membrane ion channels. So you have channels in the membrane of a neuron, which are large proteins that serve as a selective membrane ion channel. So for example, if you have a potassium channel, which is K, it only allows potassium ions to pass through. We have two main types of ion channels. We have leakage channels and gated channels. Leakage channels are always open, so ions can kind of leak through. And gated channels can only be open when a chemical binds to them, when a voltage passes through it, like an electrical charge, or they're mechanically gated and can only open if something else binds to them. Chemically gated channels open with a binding of a neurotransmitter. Voltage gated channels open in response to a membrane potential. And then a mechanically gated channel will open and close in response to some sort of physical deformation of receptors, like a sensory receptor. So here are chemically gated ion channels. They can open and close when a neurotransmitter binds to them. And voltage gated ion channels will open in response to a change in membrane potential. And we'll talk about what that means, but you need to have a little bit of understanding of these membrane channels, first of all. So when gated channels are open, ions will always diffuse or travel through them from an area of high to low concentration, so simple diffusion. And they'll also diffuse along their electrical gradients toward the opposite electrical charge. And the electrical chemical gradient is just the electrical and chemical gradients combined, and you guys don't need to know this equation. So resting membrane potential, when our nerves are at rest, a voltmeter can measure the potential or the charge difference across the membrane of a resting cell. And resting membrane potential of a resting neuron is about -70 millivolts. The cytoplasmic side of the membrane, that means inside the cell, inside the membrane, is always more negative to the outside of the cell. And that's why we call it -70, because it's referring to the charge inside of the membrane. The membrane is said to be polarized because it has this negative charge inside of it. So that's resting membrane potential. Differences in the potential or in the ionic composition can affect the potential. ICF stands for the intracellular fluid and extracellular fluid of ions will affect the resting potential, as well as differences in the plasma membrane permeability. So this is a voltmeter that's just measuring a resting neuron. And again, the part of the neuron that we're kind of looking at is the axon, and we're focused in on the inside of the axon itself. So inside of the membrane, we're at about -70 millivolts as a resting nerve. Differences as ionic composition, the extracellular fluid. So this just means that sodium ions, there are more sodium ions in the extracellular fluid or outside the neuron, and there are more potassium ions on the inside of the cell or just inside the membrane. And potassium is what will play a very important role in this membrane potential. So in resting membrane potential, this is like before we have stimulated your nerves to kind of emit a response, we have a greater concentration of sodium on the outside of the cell and a greater concentration of potassium inside of the cell. And if you're kind of getting lost already, just kind of focus, on that first of all. We're in a resting neuron. There's more sodium ions outside of the cell and more potassium ions inside of the cell. And when this neuron gets stimulated or gets electrically excited, these ion concentrations will change as ions rush into the cell and ions rush out to the cell, because these ions have a positive charge. So they're going to bring their positive charges with them, whichever way they go, and that will change the voltage of the potential. And that's how we get kind of an electrical signal to pass through a neuron, and that's called an action potential. So this takes you through some of the differences in plasma membrane permeability. Basically, the leakage channels are much more permeable to potassium. So potassium will always kind of diffuse out of the cell down its concentration gradient. This is a very important part of the cell membrane. We have the sodium potassium pump, which always helps to stabilize resting membrane potential. And what this does is it always maintains your concentration gradients of sodium and potassium ions to always try to keep sodium outside of the cell and potassium into the cell. And it does that by pumping 3 sodium out and pumping 2 potassium in, kind of at the same time. It does this pumping action, and it requires ATP to do that. So this sodium potassium pump needs ATP, needs energy to do that. Without energy, your neurons cannot create these concentration gradients, and they wouldn't be able to be excitable. So that's another reason why we need a constant supply of ATP or energy. So this takes you through kind of the different channel proteins in the resting membrane potential of the cell membrane that are establishing this -70 millivolts. The membrane potential changes if your nerves are electrically stimulated, and that occurs if the concentration of ions change across the membrane or the membrane permeability to these ions changes. And that just means that the membrane will allow the ions to pass through. We can have graded potentials or action potentials. A graded potential is an incoming signal in a neuron that operates over a very short distance. And the action potential is the long distance signal, the axon that travels the length of the axon. We're going to focus on action potentials in this chapter. So I'm going to skip over the graded potentials. These following terms you should know well. There are questions on your lecture exam, knowing very well what depolarization means, hyperpolarization means, understanding the phases of an action potential. And even if you don't know what that means now, just remember that that's kind of what you want to pick out of this chapter. And again, the workshop for action potentials will be very helpful with this. So the terms describing membrane potential changes are relative to the resting membrane potential. So when the membrane becomes depolarized, this decreases the membrane potential, so it moves it back to 0. So remember, at resting, we're at about -70. When we depolarize it, that means the inside of the membrane becomes less negative. Hyperpolarization means an increase in membrane potential or away from 0. So the membrane potential will become more negative. So it might become -90 or -100, so even more negative than resting. During a hyperpolarization phase, the probability of producing another impulse in that neuron is greatly decreased. So we can measure this. So this is a look at depolarization, meaning that the inside of the neuron has been kind of electrically stimulated. So it's no longer at resting, and the inside has become depolarized or closer to 0. So maybe -50, it might even get up to 0 or +30 if an action potential occurs. And then hyperpolarization means that the inside of the neuron gets more negative. And this is not good for the neuron because it cannot respond to a stimuli, but it's also good for a neuron in that during a hyperpolarization stage, that neuron cannot be stimulated again. So it kind of is forced to take a break or a rest. So I'm going to kind of pass over these graded potential slides because I want to focus on action potentials, but a graded potential is a stimulus applied to a neuron in a local, kind of one specific location, and it doesn't spread very far, so it's short-lived. So that's what a graded potential is, and it can go in opposite directions and it decays quickly. But an action potential, this is the principal way that your neurons send signals, and I'm going to just briefly introduce action potentials, and then I will pause and finish up a recording of this, kind of a part 2 to chapter 11, and I'll post that tomorrow. But an action potential means long distance neural communication, and this occurs only in muscle cells, which we talked a little bit about before, and also the axons of neurons. And an action potential is a brief reversal of membrane potential with a change in voltage of about 100 millivolts. So we go from about -70 to +30 in a millisecond, and that's what an action potential. It doesn't decay. Instead, action potentials travel down the length of the neuron, the length of the axon as it's sending its signal down. It involves the opening of specific voltage-gated channels. So here are the key players in an action potential. We have sodium voltage-gated channels, and we have potassium-gated channels, which will kind of have an open or a closed state depending on what part of the action potential we're in. So there's 4 main steps to generating an action potential. The resting state is when all channels are closed. And remember, at resting, we have more sodium outside the cell and more potassium inside the cell. Each sodium channel has 2 voltage sensitive gates that can either be activated or inactivated. And each potassium channel just has 1 that's closed at rest and opens during depolarization. So here's the resting state, and all our channels are closed at this point. So the nerve hasn't been stimulated yet because no ions are flowing across the membrane. So we are at resting. Again, we have more sodium outside the cell and more potassium inside the cell. So this is a resting neuron and hasn't been stimulated yet. Then we get into depolarization, and this is when sodium channels open and sodium will rush into the cell down its electrochemical gradient. When sodium rushes into the cell, it brings its positive charge with it. So because of that, the inside of the cell gets to be more positive. If the inside of the cell reaches a value called threshold, which is about -55 to -50, a positive feedback mechanism occurs, which will open all the sodium channels, and this results in a very large action potential spike, bringing the membrane potential up to about 30 millivolts. So remember, at resting, we're at this kind of -70 down here at resting. When that nerve is electrically stimulated, these sodium channels open and sodium rushes into the cell. So this normally really negative inside of the cell is starting to become more positive, and that's shown by this positive swing of the graph. This is depolarization. Sodium brings its positively charged ion with it into the cell, so the inside of the cell becomes more positive. And if we reach a threshold value, which is about -50 millivolts, we have this positive feedback mechanism, meaning if we reach threshold, we are going to generate this action potential peak no matter what, and it opens up all sodium channels. So that's what reaching threshold means. Then repolarization is the next step, and this is when sodium channels start to inactivate and now potassium channels open. So sodium channels will start to close, the action potential will peak, will stop rising, potassium channels open, and potassium exits down its gradient. Remember, there's always more potassium inside of the cell. So when channels open for potassium, it will flow out via simple diffusion down its electrochemical gradient. And when potassium exits the cell, it brings its positive charge with it. So the inside of the membrane will become more negative again, back to the resting membrane potential, and that's called repolarization. So this is the 3rd step, repolarization. We have our potassium channels opening and the potassium exiting, so the inside of the cell becomes more negative again because positive charges are leaving. And then the 4th step is hyperpolarization, and then I'll end here This is when some potassium channels remain open a little bit longer, so the inside of the membrane becomes more negative than resting state, as we described earlier, as a hyperpolarized state. So we could get into like negative 100, negative 90. This hyperpolarization state is important for cells to rest and to allow to make sure that the action potential only travels in one direction. And so that specific neuron can't be stimulated again until it gets back up to resting. So this is the hyperpolarization state. So this kind of graph gives you a good description of the 4 stages of the action potential. And I think we will pause there, and I'll kind of pick this up in a recording that I'll record tomorrow, and I'll post to our YouTube channel. So thanks for listening, guys. I'll stop the recording now. If you joined us a little late or you're still listening, you have the quiz due on chapters 9 and 11 on Sunday, as well as your first Physio X Lab. And I'm just going to go ahead and show you that right now. So your lecture quiz on chapter 9 and 11 is due Sunday at midnight. I took off Proctorio for this quiz. You can use notes. I thought that might be a welcome relief for some of you. And then your first Physio X Lab is due this Sunday, and I created a video to describe how to get to that. So before you ask me questions about the Physio X Lab, I just ask you to watch that video first, and hopefully that will explain that. So thanks for listening, guys.